Hello everyone, a very warm welcome from the Australian National University here in Canberra. I hope that you and your loved ones are keeping safe and healthy during this time. I'd like to acknowledge the Ngunnawal and Ngambri people who are the traditional owners of the land where ANU is located and from where we are broadcasting to you today. I pay my respects to their elders past, present and emerging. We're very pleased to welcome attendees from various parts of Australia and the globe, including India, Indonesia, Burma, Fiji, China and South Africa. Knowledge is power, and our weekly webinar series allows us to share and learn from each other's experience during the pandemic. I am Bridget Nielsen, a first-year medical student and volunteer for the ANU COVID-19 Peer Support Program. This is also my very first time hosting one of our webinars, and I'm very pleased and proud to be here. Joining me are my colleagues, Evangeline and Devashi, who are fellow medical student volunteers, as well as Dr. Dipti Talalaka, haematologist and program lead for the ANU COVID-19 support program. Also a special thank you to our corporate sponsor, National Capital Private Hospital. If you'd like to become a corporate sponsor, please contact international.health@anu.edu.au. Before I introduce our speaker for today, a couple of housekeeping notes. Can all attendees please have their video and microphone switched off? This helps to keep the sound quality clear and reduce interference during the webinar. I'd like to encourage all attendees to ask questions by using the Q&A button that you see towards the bottom of your screen. If you do not wish to be identified, that's completely fine. You can submit questions anonymously. You can also submit your questions at any time during the seminar. There may be some questions or evaluation polls that pop up on your screen during the talk. If so, please do select your preferred response and note that your response will also be anonymous. If you have any technical issues or questions whatsoever after the talk, please do email international.health@anu.edu.au and we will try very much to assist you. And finally, please do note that this forum is being recorded. So without further ado, please let me introduce you to our speaker. Today's webinar will be delivered by Professor William Rawlinson, who will be talking about the diagnosis and characterization of SARS, COVID-19 and variants of concern. Professor Rawlinson is a clinician scientist researching viral pathogenesis, particularly respiratory viral infections, congenital infections, and enteroviruses in type 1 diabetes mellitus. He established and oversees serology and virology clinical research programs, statewide transplant donor screening, and national quality programs for serology and biosecurity. Today, he will be discussing his research involving the utilization of novel diagnostics in the context of SARS CoV-2 variants of concern. So let's begin the webinar itself. I'll hand it over to you, Professor Rawlinson.
Thanks very much, Bridget. I'll share my screen. So thank you very much for the invitation to speak, and also to those who switched in and who are listening. Thank you very much for attending. It's a great privilege to talk to you today. Topic of my talk is going to be fairly broad around SARS and COVID-19 and a little bit at the end about variants of concern. The reason I subtitled it what I did in my year off is that as a DNA virologist, my background is infectious diseases and in virology, and my particular interest is in DNA viruses. Like many other people, I've become interested in RNA viruses and in particular in SARS CoV-2. Work that I'm going to describe, and a lot of the diagnostic work is done here on the left of your screen by the research team, in which I'm director. And the other photos are our diagnostic teams, and in particular in the mask on the right is Anna Kandelios, the senior hospital scientist in our diagnostic lab, and down the bottom right is our diagnostic team. So they're the ones who do the work, and I get to talk about what we do. I'm going to talk about a number of different areas fairly briefly. I think it's a very varied audience. I don't know all of your backgrounds clearly, so I thought I'd give a little bit about virology, talk a bit more about diagnosis and introduce the rapid antigen testing, some of the more recent research, a little bit about serology, and then I guess a bit more about whole genome sequencing. As a reference lab, we do a lot of whole genome sequencing and identifying the circulating strains, and also we use that as well as our serological work in some monoclonal antibody studies. And finally, I thought I'd talk a little bit about vaccination because that's very topical, and particularly in Australia, but I think globally that's something that's of interest. So a few definitions. It's interesting we're in a pandemic. It is something that's been predicted for a long time. And it's interesting that in many ways, the definition of a pandemic is somewhat broad. It's basically every continent being involved. And in many ways, you could argue that pandemics do occur in that epidemics, for example, of influenza cross boundaries and often affect multiple continents, although clearly we don't have a pandemic of influenza every year, and what we are in currently is largely because of the severity, the novel nature, and because of the broad and severe involvement of the populations. We're going to talk about lineages, and the Delta lineage is the current virus that's causing most infections, certainly in New South Wales and more broadly in Australia, as well as in many countries, and of course was moving from country to country. A lineage is important to think about because it has not only epidemiological issues, but it also informs vaccine responses, it informs individual outbreak responses, and so the concept of lineage is defined here. It's a sequence, so it's a genotypic definition where something's evolved from its predecessor. That is, it's a unilineal descent group. And the point about that is that these lineages are more closely related to each other than other types of SARS. We won't talk much about clades, but that's basically what's called a monophyletic group. That is a group of organisms that are composed of a common ancestor. This is the sort of timeline that we've seen in Australia. A lot of politics has taken over these kind of diagrams. I talk from experience and from what I know personally. The early clusters of pneumonia of unknown origin were reported by China in late December, although subsequently it does appear there were cases probably back into mid-November. The cluster was clearly related to a seafood wholesale market in Wuhan in China, in Hubei province, and subsequently the first death was recorded. And on the same day, Eddie Holmes, who's an Australian, uploaded the sequence which Professor Zhang Zheng his laboratory had determined. And that sequence was uploaded and very rapidly allowed us to develop diagnostic tests for what subsequently was called SARS coronavirus 2, a virus causing the disease COVID-19. First case outside of China was early in mid-January, and we began testing in Australia in my laboratory on January the 27th. At that stage, we'd only had one recorded case 2 days prior, and so our nucleic acid test, our PCR test, was based upon artificial templates because we hadn't seen cases at that stage in Sydney where I practice. These are the figures for Australia. Much of my talk is Australian focused because that's what I know about, although I think some of the lessons that we've learned from other countries, and hopefully some of our lessons are useful to some of the people listening tonight. In Australia, currently there's 87,000 confirmed cases. That's gone up very rapidly over the last 2 months. That's about 0.35% of the population. And one of the really interesting things that is occurring is exactly what we would predict from a pandemic. We're seeing the virus becoming more transmissible and less lethal. That is, the Delta strain, the B.1.617.2 strain that we currently see is transmitting much more easily. It has an R0 of about 5.5 compared to the R0 we saw with the original strain, which was an A strain from Wuhan, and that was an R0 of about 2.5. So about 2.5 people being infected by one infected individual. We're now seeing more spread of the Delta strain, but if you do crude death rates, our crude death rate is more than halved. Now, we're not at the end of the current outbreak, and it may well be that rate goes up, but it is very interesting that the sort of predictions that we made many years ago for pandemics and for respiratory pandemics are the sort of appearance we're seeing now, and hopefully that continues. That is, the number of deaths per case decreases. So now I'm going to talk a little bit about the virology, because I think it's really important to understand the virology and the basics of pandemic viruses in order to understand what's happening and what's likely to happen and how we should respond. There's no doubt that SARS coronavirus 2 closely resembles bat coronaviruses. It's very similar to SARS coronavirus we saw in 2003. There were no cases in Australia. The sole case was a German tourist who returned and in fact was diagnosed in Germany. So SARS-CoV or SARS-CoV-1, if you like, was not seen in this country, although it was seen particularly in China, Hong Kong, as well as in Canada and a number of other countries. The last case of SARS-CoV, of course, was 2004. It was a laboratory-acquired case. And so SARS-CoV2 came along somewhat unexpectedly, although in many discussions subsequently, it did seem a likely occurrence. SARS is one type of coronavirus. The other 4 types that infect humans, NL63, 229E, OC43, and HKU1 shown here. And it's believed that these have predominantly come from bats. It's interesting that OC43 probably was the cause, or possibly NL63, but probably OC43 was the cause of a pandemic in 1896, which was attributed to influenza, but increasingly may well have been associated with that coronavirus. And these 4 human coronaviruses, although originally zoonoses, are now spread as basically the common cold yearly between individuals. They're very closely related to SARS-CoV2, and of course, as I mentioned, very closely related genetically to bat coronaviruses. The diversity of coronaviruses means that we are able to construct phylogenetic trees, as shown here. And this is a time-measured phylogenetic tree where divergence is estimated going back. And this tree goes back as far as 1200 up to 2020. And essentially what this shows, just to summarise a lot of work, is that the pangolin strains of coronavirus, the bat strains of coronavirus, and the human coronaviruses we're seeing now are very closely related. The whole question of the origin of SARS-CoV-2 is both a political as well as a medical issue, and there's a lot of argument. The science at the moment is that coronavirus we're seeing in humans closely resembles a bat coronavirus. It's referred to particularly as this RATG13. It also closely resembles pangolin, and these trees that you see here very much depend upon how you draw them in terms of how closely related they are, but these are very closely related. Whether the pangolin as a mammal was an adaptive animal for transmission into humans, whether it went from bats to pangolins to humans or from another source to pangolins to humans, it's still not clear. And the genetics really don't answer that question at the moment. The other politics, of course, is around where it came from, and I'm not going to go into that tonight, except that epidemiologically, we saw most cases arising initially in Wuhan, closely related to a wet market. And in the early days, this is a publication on that. The cases before January the 1st tended to be slightly younger. They were roughly equally distributed between men and women, and they were very closely related to individuals around the seafood wholesale market in Wuhan, in a place called Huanan. It's interesting, of course, as you went further and later into January, the number of cases associated with the seafood market as a proportion decreased as the virus spread more widely, and disappointingly, the number of infected healthcare workers increased. And in fact, some of the original deaths were in healthcare workers, particularly an ophthalmologist who clearly had early exposure and sadly died. A number of those cases were quite young people. This is a quote I came across fairly recently, and I thought it was interesting because we now accept that we wear masks. We now accept that we're in the midst, or probably the midst of a pandemic. We're certainly in a pandemic. It's interesting that we have adjusted and many things have changed in our lives. And yet, of course, what we are doing now is quite different to what we did 2 years ago. And yet we've adjusted to that. I'm going to talk a bit about diagnosis because that's what I do. I direct a reference laboratory. It has research components. It has service components. And we do a lot of molecular testing, and we do a lot of serological testing. And we do a lot of public health work at a policy level. The viruses we're talking about, the coronaviruses, as I mentioned, there are 4 known coronaviruses which are established in the population for at least 100 years, and they cause around about 1 in 8 common colds. Of course, the other viruses that we see, like influenza, can also cause severe infection, but they've been present in the population and circulating for many years. Things like rhinoviruses, things like parafluviruses, metapneumovirus also cause large numbers of the common cold and typically not associated with poor outcomes. I mentioned NL63, 229E, OC43 and HKU1 because they are cause of the common cold, although interestingly, papers in the last 6 months have suggested there are still quite severe outcomes from these in some cases. Respiratory viruses are typically those that cause major respiratory infections, but things like measles, for example, of course, can also cause pneumonia, but they're not what we're more concerned with tonight. SARS-CoV-2 resembles very closely SARS-CoV, as I mentioned. It's also closely related to the circulating MERS-CoV, which is still circulating, particularly in the Middle East, and is more associated with animal to human transmission rather than ongoing human to human transmission. And of course, we now have SARS-CoV-2, the causative agent of COVID-19. One of the reasons I call this what I did in my year off is because basically my laboratory, like many in the world, and certainly many in Australia, has basically swapped from a general virology laboratory where we did HIV, Hep C, herpes virus diagnostics, doing about 1,000 tests, molecular tests a day, and about 2,000 to 3,000 serological tests a day, to now doing those tests, but also doing large numbers of tests for SARS-CoV-2. The top panel with the pink shows the number of tests done nationally in Australia. This is on a daily basis, and these are the average numbers. So the testing rate's gone up particularly considerably as the number of infected individuals within Australia, and particularly in New South Wales, and more recently in the ACT and in Victoria, have increased. Down the bottom left are the number of tests done in the Australian Capital Territory in Canberra, and this really increased in the second week or so of August, again, as the number of cases increased in the states around Canberra. And in fact, more recently, as the number of cases in Canberra have also increased. As a diagnostic laboratory, we had to adapt to this very quickly. As a research laboratory, we were fortunate to have a number of molecular biologists who had PhDs who worked with me or had come to work with us as postdocs. And those people really assisted us in January of last year in setting up, as I mentioned, assays prior to the first cases occurring. The other point was that a number of our processes, things such as the pre-analytical, how we handle specimens, the post-analytical, how we're going to get these results out to people, all had to be rethought because we went up from something like 1,000 molecular tests a day to around about 4,000 to 6,000 SARS tests a day, and we had to continue with our other testing. So like many laboratories around the world, we had to change a lot of our processes. So the point about this diagram is really to say we considered a lot of things. And in fact, for example, our research staff participated early on in much of the development of assays. We had a lot of international collaborations, and we particularly collaborated with people in Hong Kong, Leo Poon and his laboratory in Hong Kong, who developed early on assays because they had more experience because they'd been in the middle of the outbreak in 2003. And so we had the benefit, particularly early on, of other people's expertise and other people's help. We had the benefit of our own research and our own capacity. And so what we did was went from a smaller laboratory and a public health laboratory to employing many more people. We went to do things like having dual teams. So there was the white team shown up the top right and the blue team shown down the bottom left. These people don't cross over at all through the day. They don't share tea rooms. They don't have any unmasked or uncontrolled interactions, so that they if one of them develops SARS-CoV-2, then that team can be taken out. They can be appropriately quarantined, and the other team takes over. We have a third team off-site, and they can come in if necessary. So staffing was probably the most important thing that we did, and those processes changed considerably. The other important thing was resources, and of course this is how we did it. A number of laboratories do it in many different ways, but this resembles what most of the reference laboratories in Australia did, which is to increase their staff, to increase training, to also increase our equipment, and this is physically the equipment we currently have, or we had in 2020 going forward. These are SAViD Gene Experts. This is a Roche 6800 high throughput machine. This is an Australian machine. It's called Genetic Signatures GS1000. We increased our platform such that we had redundancy and we were able to handle up to 7,000 or 8,000 specimens a day. Now, just to put that in context, some of the private laboratories handle 12,000 to 15,000 specimens today, and of course, laboratories in other countries handle many more than that. But we also have public health responsibilities and we have policy responsibilities. And so this really allowed us to back up what we were telling people in a policy setting with what we were seeing on the laboratory bench. We have different platforms, as I mentioned. Some of them take 5 hours. This current platform with the genetic signatures is currently going down to 3 hours. The Roche platform takes about 3 hours to process specimens, and rapid platforms like the Lineage, or in this case the SAViD Gene Expert, take about an hour. The other thing we did was pool specimens. We started very early on pooling because, as many of you will recall who were involved with this, in January 2020, we didn't know whether we were going to actually have resources. We didn't know if we were going to have access to testing platforms, to testing reagents. And so we started out with pools of 4. That allowed us to quadruple the utility of the consumables we had. So this is what we do. As I mentioned, everybody has a different process to some extent. As I mentioned, we commenced now 19 months ago. Initially, we did in-house assays. This was based around a Hong Kong University assay, as I mentioned, that Leo Poon and his laboratory kindly shared with us. We do have employees who work with us and a couple of students who worked in his laboratory, so we knew them very well. And then rapidly, the commercial assays became available. And as I mentioned, we had several different assays. They were really basically to avoid things like reagent shortages. The mean time between failures for equipment decreased. So as we utilised the machinery more, unsurprisingly, it broke down more. And so we specifically aimed for the use of 2 or 3 platforms, including one provided locally. And we early on engaged with processes to reduce staff stress, to reduce illness, because of course during winter things like influenza and common colds arose. Interestingly, in 2020 and again in 2021 in winter in Australia, we've seen virtually no influenza with the mechanisms that have been introduced to reduce SARS transmission. So it's interesting that that's had that effect, although we've still seen common colds. And so this is just a schematic of the outbreak in our experience from January 2020 up to a couple of months ago. And these are the 3 major outbreaks that we've seen within Sydney and within New South Wales. Initial one around April, May of last year. Another one at the end of last year, which was very short but very high. And most recently, the most numerically severe, and in terms of admissions to hospital, the largest number of admissions, although as I mentioned, fortunately not reflected in increased numbers of deaths, not reflected in the same increase in deaths in those cases as we saw previously. The most recent outbreak has been the largest we've seen so far. The aim of our testing was to provide a test initially within 8 hours. That blew out to providing a test within 24 hours, and we basically stuck to that since the testing has begun in our laboratories. We have a very close relationship with our public health unit, and one of the things I think that has been done well in Australia is that a lot of public health units are closely related to testing laboratories. We have an ongoing relationship with them. We have exchanges. We've educated them and they've educated us about processes, and that's been enormously beneficial in this setting. There was early on discussion of analytes like stool or urine, blood. This is not a viremic virus. You can find it in stool and urine, and that's most useful in testing of sewage. We're involved in whole genome sequencing of sewage-detected virus, but the testing is actually done through water companies. Things like the supply of swabs became important. We have access to a PC3 laboratory, so we do our virus culture there. that also allowed a lot of things when strains developed or new strains developed for providing reagents that we could use in diagnostics. And so what we've seen is the rapid introduction of existing technology, of new technology, the quality assurance programs. I think Australia certainly introduced the first QA program nationally in the world. I have just published the results of that. We've done QA programs for whole genome sequencing as well as for detection. There was an enormous amount of data sharing, and that goes on, both at a genomics level, but also in terms of diagnostics and other processes. And there's been a lot of governance issues because, as you might imagine, there has to be some coordination of all of this, and I think each country has addressed this in different ways, but one key feature has been the emergence of the use of genomics as an important feature of our response. Rapid antigen tests, I think, are something that I'll deal with briefly. A feature of some countries' responses, particularly where there has been large numbers of cases, and I'm thinking of countries like the US, where rapid antigen tests have suited their population and their processes. In Australia, they've been less introduced or less used. On the right's an example of a recent investigation which did use rapid antigen tests. There are, in Australia, through the Therapeutic Goods Administration, 136 kits registered at this time, so there's a lot of choice. They are used, they suffer from sensitivity issues. A sensitivity of 93% we would regard as inadequate for a nucleic acid test, but they have enormous benefits in terms of use in remote site. There's questions and there are places where they're used, for example, for self-testing. When I discuss rapid antigen tests, it's important that some of the rapid serological tests, which were introduced in Australia in about early to mid 2020, are a different process, and I'm not going to discuss those. And so what's happened most recently where I work is that frameworks for provision of rapid antigen tests in clinical settings as well as in non-clinical settings, such as in companies within aged care services. And on the right, this is an aged care service example where they undertook rapid antigen testing, has been really a way to regulate these and optimise their use. This is from actually the University of Melbourne Vet School, but this is a nice way of assessing how assays work and how well they'll pick up a population, because you can actually adjust the sensitivity and specificity. And for all these tests, we have well-defined specificity and sensitivity. And then you can give an estimation of the pretest probability of disease and then decide on things like positive predictive value. And for some of the rapid antigen tests, the positive predictive value is very low. The negative predictive value is much higher. And so they have been used in settings, as I mentioned, such as aged care, where the pretest probability is quite low. And really what people are looking for is a rapid and local diagnostic. So they have a role. They have problems. There are many, as I mentioned. There's 136 listed on the Australian Register of Therapeutic Goods, or the ARTG. And this is one example. This is a Binax test. And this is just comparing on the left the CT values, that is RT-PCR result, with the Binax estimate of amount. And they roughly correlate with nucleic acid tests, and they cluster well with positives and negatives. But, as I mentioned, in a low pretest probability, for example, the positive predictive value is not good. And so we basically are largely doing nucleic acid tests currently, although I think in going forward there will be changes in the way we conduct testing. This is a Cochrane review. This was done last year and was a very good review at the time. It's now out of date because things have developed so rapidly with this. Serology as a test has been around for over 100 years, and it's interesting because it has become useful and it's remained useful in various settings. In most settings, we're measuring antibody using enzyme immunoassay, or EIA, but there are also rapid antibody tests which I'm not going to talk about, as well as other platforms, including immunofluorescence. There's been recent publications in the last couple of weeks looking at duration of serology, seropositivity, and certainly post-infection seropositivity for 12 months and longer is being seen, whereas post-vaccination seropositivity of sort of 6 months and slightly longer is being seen. The assay performance is very much determined by what marker you use and what target antigen. Many of them are directed against spike antigen, which of course is the component of the vaccine. And so you can use a test which measures spike antigen, measure vaccine response, and then compare that with a response, with the serological response to, say, the N protein, the nuclear protein, as antigen, and use that as a differential. So if somebody is spike positive and nuclear protein negative, you presume it's vaccine derived antibody. If they're spike positive and nuclear protein positive, you presume it's natural infection. It's not as accurate as that, of course, and we looked at this using microneutralization and just published this a couple of months ago. And as others have seen, the assays for serology are reasonably good. But things like decaying antibody, especially neutralizing antibody, things like the sensitivity for infection and just the inaccuracy of some of the assays means that, again, they have to be interpreted in the epidemiological setting. And these are some of the data which Greg Walker published, as I mentioned, just a couple of months ago. But this is what's seen. There's a good correlation between the reference assay, which is microneutralization, which is a functional assay, and many of these EIAs. And these are some of the tests from Abbott, Cobas, Euroimmune that we use in our laboratory. And it's not one-to-one, but it's reasonably good. And so serology is useful in a number of settings. We now have quantitative assays and we do have very good EIAs, so they are useful tests. We do a number of research programs and there's a lot of work on rapid near patient testing and using other forms of assay to improve local testing. And I think that's going to be the big change that we're seeing going forward. So I'll talk a little bit about characterizing the virus from whole genome sequencing, because I think this has become one of the major public health interventions that we've got. This is the old-fashioned Broad Street pump picture, deaths from cholera related to particular source of infected water. Traditional epidemiology, up until a couple of years ago, very much standard. But now what we have, of course, is the availability of genomic epidemiology. And it's important to point out that this is a component of the epidemiology and of tracing spread. I think it's actually becoming the most important component, but it's very, very important to realise it's not the only component, and cross-sectional and case control and cohort studies remain extremely important. And this is really probably when genomics became most used. This is the Ebola outbreak in West Africa a couple of years ago, well now 6 years ago. And the epidemiology really was supplemented very much by genomics and very much showed spread of the Ebola virus in Western Africa. These are from GZAID, which is the database that we mostly, there are many databases that this has become the primary database for downloading our SARS CoV data. This was a shot from earlier this year. We're actually now up to over 2 million genomes, and so it's just exponentially going forward. And these are some data, and there's not the time to go into these in great detail, so they're really illustrative. But the point is that there is a complex evolution of the virus going on, which is related not only to changes in certain sites, not only changes, for example, in the spike glycoprotein, but changes throughout the genome, but also recombination, and possibly recombination between human and bat coronaviruses. And there's been some work just in the last 3 months that suggests that's much more common than we believed. That in fact bat to human transmission has been much more common than has been observed, because many of these infections have been non-lethal or non-severe and have really not led to ongoing human-to-human transmission. So recombination as well as site-directed mutation is extremely important, and you can map not only bat viruses but also human viruses along them. There's been a lot of emphasis on the spike domain, which is a glycoprotein, and there were several characteristics that were thought to be unusual, particularly the presence of furin sites. However, the sites that we've seen in human viruses are also identified in bat coronaviruses, and it does seem that the close genetic relationship between those two is quite clearly bat to human transmission. The other thing that's happened to me as a diagnostic clinician has been that we want it now. As I mentioned, we aimed as rapid delivery of results to reduce transmission, but alongside that, and particularly what have been called the variants of concern, that is genetic mutations that are associated with high levels of transmission, the demand for rapid genotyping and determination of transmission events locally as well as internationally has just spiraled. And the we want it now has led to people like my laboratory providing genomic data on outbreaks within about 8 hours using technology that we developed last year, and which is freely available. Why do lineages? So why do we have, alpha, beta, gamma, delta, lambda, mu? What's driving the spread of lineages? And it's interesting because it may be that mutations are actually providing a fitness advantage, particularly in terms of increased transmissibility. It may well be that a lot of that's within the spike protein, but it's not only within the spike protein. It's also within others, particularly the nuclear protein, and there's increasing evidence that that's associated with aspects of transmissibility, and it makes sense. The other thing, of course, is a founder effect. Once you get a more fit and a more transmissible virus, that will tend to become founded in the population spread. And we believe that's what we've seen with Delta, which is the current strain. Strains, of course, were named after the place they were first recognised, not necessarily the place that they first came from, but subsequent to that, their lineage names have been used. And sadly, for a virologist, we now call them alpha, beta, gamma, delta. I find that much harder because the lineages, of course, are quite informative, because what was formerly called the UK strain, the B.1.1.7, you can immediately see is related to the B.1.617.2, which was also called the Indian strain. As I mentioned, this is where they're first recognised, not necessarily where they first arose. So where we are now, we've got a lot of information about the lineages. They're based on what's called the Pangolin lineage classification, and the new WHO label, which is a simpler label to say but is not as informed, is now being used. And there are large numbers of lineages circulating. Many of them have similar characteristics, particularly the later ones in terms of increased transmissibility, but they haven't all become established. One of the bottlenecks in understanding local outbreaks was the turnaround time for sequencing, which in some cases was up to a week. Using some of the more recent Illumina equipment came down to about a day or two. It's a very accurate, very good system, but there are other systems, and one of the ones that we looked at was the Oxford Nanopore MinION, which is less accurate but can be much more rapid, and in fact can give results within an hour or two. And so this is some work we did. Other people have done similar work subsequently, but basically this was using nanopore sequencing to determine lineages and therefore describe an outbreak much more quickly. And we now determine lineages the same day for urgent outbreaks. For non-urgent ones, we use the Illumina platform and determine them the next day or a couple of days later. The problem with the ONT system has been the error rate, although you can actually take account of that. And by accumulating more data, by running them slightly longer, you can accumulate enough data to get accuracy approaching the other platforms. There are swings and roundabouts about different whole genome sequencing platforms, and it's very important, and we're just publishing this, and it's known, but it's really quantitating how important all these different things are, how you process your raw reads, what your thresholds for calling a base are, how you map the genome and which algorithms you use, how you call a variant and what thresholds you use. all of these things are really important in how accurate your sequence is. And in fact, just to give you an example, fairly minor changes in sequence can give you quite significant difference. The lineages remain similar, but the lineages that we see, and this is one example, one of which really just minor changes in the sequence led to a significant change in our understanding where this sequence might have come from. The B.1.260 being very localised predominantly within the Europe and the Middle East, whereas the B.1, of course, being a precursor lineage and being much more broadly arising and initially being seen in an Italian outbreak in 2020. The next steps are things like the use of looking for co-infections and using that to determine prognosis, doing things like using the whole genome sequence to inform the construction of new monoclonal antibodies, which we do with Daniel Christ. I guess looking at how these things inform how we might control outbreaks, but increasingly how we might vaccinate and prevent infection. As I mentioned, my focus is Australia, so these kind of data are available for all countries internationally, but this is just to give you an idea of the context in which we work. Australia's vaccination rate until recently was not very high compared to the rest of the world, but increasingly over the last 2 months with the current outbreak, there's now about 75% of the population who've had one dose. Although the fully vaccinated rate, and these are data from, I think, yesterday, is still down at 38%, but increasingly Australia is achieving the sort of aim of 80% vaccination rates. Whether that's the right number or not is a completely different discussion. There's many vaccine candidates. I'm not going to talk about them all, but this is updated today to give you, and the size of these green circles give you an idea of the number of vaccine candidates. And there are currently 215 trials recruiting. 18 of those are within Australia, and there are 34 RCTs currently underway. I won't go into all the candidates, but the predominant candidates are RNA-based vaccines, so predominantly mRNA vaccines like the Pfizer and Moderna vaccines, followed by protein subunit vaccines, although viral vector vaccines are being developed, and I think they're going to be particularly interesting because there's going to be a lot of discussion about their utility and their comparison with the mRNA-based vaccines, and also the fact that they may be able to represent larger numbers of epitopes. These are the predominant ones seen in Australia: the AstraZeneca, Pfizer vaccine, so protein and an mRNA vaccine, and then Moderna and the other vaccines coming along like Novavax. All of them actually are based on the spike glycoprotein at the moment. But again, there are other whole vaccine candidates that are being used elsewhere. There are a lot of misconceptions. I deal with a lot of this in my work now, as we have an aim to approach as close to 100% vaccination as possible. Things like the similarity between a protein associated with fertility and the spike glycoprotein, the fact that the Delta strain spreads despite vaccination, but there's quite clear evidence that double vaccination in particular prevents severe pneumonia and death, and things like around the rush development. My comment is these are the largest vaccine trials ever seen in the world, involving many times the number of candidates that have been involved in previous vaccine trials. So look, that's all I'm going to say. The other thing that, of course, the pandemic has led to has been social distancing. And for those who have a copy of The Beatles' Abbey Road, we've redesigned the cover. The diagnosis has been the key focus of most of our work until the last sort of 6 to 8 months, when vaccination has become the focus. And it's interesting how important genomics has become, and how important that those lineage data and those whole genome data have become in not only things like evolutionary dynamics, which is interesting research, but local control of outbreaks and the production of vaccines, and interesting how quickly those genomic data have been translated. So as I mentioned, I'd like to thank the people who actually do the work. I'd like to thank the support we get from the Commonwealth and New South Wales ministries, And close with a quote from Fred Sanger, who, of course, received 2 Nobel Prizes, the first for protein sequencing of insulin and the second for developing DNA sequencing, which is the basis for everything we now do in genomics. And I like his quote that through art and science in the broadest sense, it's possible to make permanent contribution towards the improvement and enrichment of life. And it is these pursuits that we students are engaged in. And I really like this because I worked in his laboratory. He sadly died a few years ago. He was a fine human being, and he was always a student of what was happening. He was always learning, and he was always willing to make corrections in seeking for the truth. And I think that that's what we have to continue to do through the politics and through the difficulties and through this pandemic, because that's what's ultimately going to lead to the best outcomes for all of us. And so that's why I like this quote from Fred Sanger, because he regarded himself as a student just like the rest of us. So thank you very much for your attention.
Thank you, Professor Rawlinson, and I agree that's a fantastic quote. We do have a few questions from our attendees. I'd like to invite you to answer them. Okay,
so I'll read them out, I think is the idea, and I'll attempt to answer them. The first one's, what do you think will be the role of rapid antigen testing for case detection in routine clinical workflows? It says Evangeline Wong is going to answer this question live. Maybe I'll answer it, and then if Evangeline's got an answer, she can answer it. That'd be good. The reality is, I think in a low incidence, low prevalence situation, which is what we've largely seen in Australia, I think that they're going to be useful in some settings where access is difficult. The example I showed was aged care setting. I think as you move towards higher incidence and more critical issues, not necessarily in a personal sense, but in a public health sense, for example, in a clinician who may be running an ICU, I think that rapid antigen tests in those clinical workflows are going to be more problematic. The other setting that people are using them in now is to have a rapid antigen test collected alongside a PCR assay, and both of those assessed at the same time. The benefit is the rapid antigen test gives you an answer in half an hour on site. The PCR test is then in process and gives you a later answer. So you can act on something that you know is not as good, but is giving you a reasonable educated guess at the time, and then later on get the definitive answer. By doing them in parallel, that later on becomes sooner rather than later. Were you going to say something, Evangeline? I wasn't sure.
No, she's just...
I was just highlighting it, highlighting one of the questions that you will be answering. Yeah.
Are the viral cultures done routinely or only on those on RT-PCR positives? Viral cultures are not done routinely. They have to be done in a PC3 laboratory. They're expensive. They're useful in release from ICU. So what we've seen and what we're just writing up at the moment is we've seen patients who are brought into ICU, typically with the previous strains, the B.1 or the A strain. We would see those patients getting better or becoming critically unwell, and in some cases dying over a period of a week or two. And that's what you saw early on. And for example, if you look at recent discussions with Boris Johnson, he was a classic example of that. He felt pretty well, he was doing okay, and then he crashed about day 5 and required more intensive support. And that was a classic thing we saw with patients early on. What we're now seeing is that they're admitted to ICU with the Delta strain. They're unwell, they develop pneumonia, but then they get a bit better. They get lower viral loads, so they're apparently controlling the virus. But then after a week or week and a half, they start to develop higher viral loads, they get a bit sicker, and then they get a bit better. So we've had a patient out to 23 days who's actually in ICU and is unable to be released from ICU because they're still infected and they're still unwell. That's not what we saw last year. And in fact, the ICUs around the country, certainly in Australia, now have a large number of these patients. And some of these patients, in order to release them, you need to do viral culture on. So that's the setting where we've used it, because we can't use molecular testing to tell us whether they're transmitting virus. They're stuck in ICU. The ICUs really unable to take more patients. And so we're determining on the basis of viral culture of those patients whether they're transmitting live virus, and if they're not, they're then being able to be released from ICU. How did we go about NATA accreditation in the light of rapid change in technologies? Really good question. They're all good questions. So basically the question for us is, as a accredited laboratory which has certain processes to obtain that, how do we rapidly change things like pooling, which you don't do in routine diagnostics usually? The first thing is we engaged very quickly with the quality assurance program. So 10 years ago, I set up a quality assurance program for the Commonwealth for agents of biosecurity. And what we did within that program is to start a QA program for molecular testing for SARS. We set that up in February of 2020, so within a month of the first cases, and so laboratories participated in that. That program's now been through another thing, I think 3 or 4 iterations. We did the same for whole genome sequencing about June of last year, again through the RCPAQAP, and that provided quality assurance data. We also engaged early on with the Therapeutic Goods Administration and with the National Association of Testing Laboratories, of NATA, and we asked them what they wanted. We asked for some leniency because I don't know how much you remember of that time, but for me, there were questions about whether what the mortality was. Was it going to be, accrued mortality of 1%? Was it going to be 50%? We really didn't know. We really didn't know as much about transmission. Was it fomite driven? Was it aerosol driven? So there were a lot of questions. And so, in fact, the engaging with the regulatory authorities in a realistic manner and saying, as a laboratory, we have a responsibility to do this in order to save lives, but we want to do it the best way we can. That allowed us to go ahead with things like NATA accreditation. That's been very useful. We're sort of back to square one again now. Things are really, the testing that we do and that all laboratories in Australia do is now NATA accredited for SARS, and most people have moved away from in-house assays. Those still doing in-house assays have had them accredited. The next question, any issues with serology cross-reactions like we see in flavivirus? The short answer is no. We did a lot of assessment of sera early on. Many people did this with microneutralization and also looking at sera from patients infected with non-SARS coronaviruses, so the OC43, NL60, so OC43, NL63, 229E, less HKU1. There's not much HKU1 in Australia. And we didn't see serological cross-reactivity at all. I think that's because the spike glycoproteins are very different, and they are very different viruses. One question, I guess, is are there people who are serologically cross-reactive because they've acquired bat viruses without knowing about it? We don't know the answer to that in Australia, although there have been some studies in Asia and where people have been working with bats, and there's possibly some cross-reactivity in that setting, but probably not enough to give you false positives in a clinical sense. So it's very different to flaviviruses. What's the maximum number of samples that we've pulled together, and how do we validate that? So we currently routinely pull 4 just because it's a convenient number with the pre-test likelihood of positivity. We've done up to 12 where we saw some reduction of about a log in sensitivity, and we have pulled 8 where we see a slight reduction in sensitivity. And if you say for those who sort of read RT-PCRs, when you look at CT values, which are an approximation of quantity of virus, 3 CT values is about a log. With pools of 4, we see about a 1 CT change. We validate that simply by comparing spike samples. So we have both expressed plasmid DNA of specific targets as well as whole virus DNA. We've done limit of detection and quantitative assessments of pools versus single versus CT values. And we did that early on. That very clearly showed that pools of 4 worked very well. And we then did some live when we had large numbers of positives, submitted those data to the TGA and to NATA, and they accepted that that was adequate performance. And from the sort of end of the bed, from the general kind of oversight, we don't see patients with significant infections who are missed by pooling. We occasionally see patients with very high CT values. If we assume for many assays a cutoff of around a 40 being the positive negative cutoff, we see patients sometimes who on a pool will have CT values of 39 on one of 3 targets, but on singles might have CTs of 38 on 2 targets, for example. So we do see that those are not patients who transmit. We don't see transmission typically above 30 CT of 35. The next question is, what's the value of testing antibody levels as a marker of vaccine effectiveness? Really good question. This is something that everybody's struggling with at the moment, and I think over the next couple of months it's going to be increasingly used. The short answer is it's in the general population of little use. As we go to next year, when vaccine effectiveness is going to have waned, antibody response is going to have waned, and potentially vaccine effectiveness is going to have waned. We really don't know this just yet, although there are some data from the Northern Hemisphere. We may well be using antibody levels as a guide to revaccination. I think that's unlikely given the number of people who are going to be vaccinated, that is in the millions. Where it's more going to be used is in the small subpopulations where vaccine efficacy is reduced, so the immune compromised. And for example, the FDA has recently been considering, and transplant groups, both in the US and Australia as well as in Europe, and I don't know, there may be others, have been considering 3rd doses of the vaccine for immune compromised patients. These are a population of individuals who, say, have received organ transplants or hematopoietic stem cell transplants. These are patients who have had large numbers of procedures, who are at risk not only of infection but of severe infection and death. You see higher lethality of the Delta strain, certainly in hematopoietic stem cell transplants, slightly higher, not as high as we had expected to some, but you do certainly see severe illness. And so the consideration in those patients may be whether they have adequate neutralising antibody, and if they don't, then giving them the 3rd dose. If they do, potentially not vaccinating with a 3rd dose. So I think antibody testing is going to be a much better marker of vaccine effectiveness in those at-risk populations. The other setting it's going to be used in, I think, in going forward will be in looking at individual strains. We've been talking about the Delta strain a lot because that's what's circulating, but there are strains coming along. And as I mentioned, with recombination occurring within SARS CoV2, there's going to be a lot of people who will need to consider whether you're going to be long-term vaccinating one virus or whether you're going to be vaccinating for different viruses on a yearly basis. And the reality from my point of view is that it'll be very similar to influenza. You're going to be vaccinating people every year or possibly every 2 years. You're going to be using a different vaccine, and you're going to be reformulating it based around what the circulating strain is. Now, that's not known. That's not something that we have a lot of experience with yet, but it's kind of predictable, and that's where serology is going to be very useful. Could we have that quote from P.G. Wodehouse again? To the thinking person, nothing is more remarkable in this life than the way in which humanity adjusts itself to conditions which at their outset might well have appeared intolerable. And that was made in London in 1926, and I think it is a reminder that this is not the first pandemic that the world has experienced. Have we tried comparing the receptor binding domains of bat CoV and human CoV? Yes, certainly. The receptor binding domains of, for example, the RATG bat coronavirus has been very closely compared with human coronaviruses. They have, can I say, they have similarities and differences. They have similar structures. They have similar folding in that there are key residues, but those key residues differ between those two. The other thing about a lot of those studies, and we've been involved with some of them because of our work with Daniel Christ and the Garvan Institute and the Kirby Institute on monoclonal antibodies as therapeutics, that the problem is that you express these proteins and so you express your spike protein and then you try and express the receptor. Once you overexpress the receptors, for example, the ACE2 receptor to which the spike glycoprotein binds, then you get changes in the structure with that overexpression. It's very important to maintain them in the native configuration. And so that's where you need to really look not only at the in vitro model and the overexpressed ones, but also those expressed in cell lines. And one of the things that we do at University of New South Wales is we have a collaboration where we look at organoids. So Shaffer Waters has a very nice respiratory organoid where you can actually create effectively a microscopic alveolus and a microscopic lung, which you can progress and you can then infect. And so you can look in real models that is as similar as you can get to a human lung at how the human coronavirus is binding, how SARS CoV2 is binding. We haven't done the same experiment with bat coronavirus because that's very difficult to do in Australia. It's ethically quite difficult for obvious reasons that you need PC4 and dealing with both of those viruses in high concentrations within a laboratory requires a lot of regulatory and ethical oversight. It's an interesting question, but it's not been done within those lung models. It's been done only for the human coronavirus. And the other thing we're doing at the moment is to look at comparing those SARS-CoV-2 with the other OC43 and 229E coronaviruses that we grow up in the laboratory. So, Bridget, there's no open questions.
No, I think you've answered them all terrifically. Thank you. Thank you again, Professor Rawlinson. I'm just going to open a poll and I invite all our attendees to please submit a response. And again, I just want to really thank you very much for your talk this evening. I'm sure our live attendees all enjoyed it very much, enjoyed your quotes, and I'm sure those watching the recording post-webinar will also find this talk very educational and informative.
It's a great pleasure. Thanks very much for having me.
Thank you. And thanks to our wonderful audience as well for joining in today. Thanks. Okay, so I'd just like to leave a quick note before we finish, is that our upcoming webinar is on the Wednesday, the 29th of September, which will be delivered by 4 esteemed speakers from Australia, India, Singapore, and Fiji on an incredibly important topic titled Should I or Shouldn't I? Challenges of health professionals accessing help for themselves. This session will discuss the psychological impact of the pandemic on healthcare professionals. The international panel will discuss ways to self-care as well as seeking help when needed. If you're interested, please do feel free to register by visiting the ANU COVID-19 Peer Support Webinar Series website. Thank you and have a good evening.
Thanks, Bridget. Thanks, Dipti.
Thank you, Bill. It was an awesome webinar. Really appreciate you joining in.